Hello, my name is Arjun Garapaty. Before I start my presentation time, I would just like to note that my internet connection has been a little bit unreliable and it's caused some lags in my audio. So in an effort to reduce those, I've turned off my video and I'll turn it on towards the end of my presentation. But for now, in order to avoid those lags, I'm keeping my video off, if that's all right. So without further ado, let me begin my research presentation. So hi, as I mentioned, my name is Arjun Garapati. I'm competing in the high school division, and the title of my project is Design of a Novel Inhibitor for the Cdc14 Enzyme in Fusarium oxysporum. So in recent years, an unprecedented number of fungal diseases have jeopardized food security on a global scale, with wider implications for human and ecosystem health, which have been further exacerbated by population growth and climate change. This has prompted the development of synthetic fungicides, a group of pesticides that target fungal pathogens. One of the most common of these chemical approaches is the development of molecules which inhibit biological processes necessary for fungal growth and pathogenesis. My project focused specifically on the development of a novel inhibitor for Fusarium oxysporum, a pathogenic fungal species which infects various economically and nutritionally crucial crops worldwide. Despite the severity of its effects, treatments for F. oxysporum have largely been uninvestigated. So for this project, I isolated the Cdc14 family of protein tyrosine phosphatases as a target for F. oxysporum inhibition. I deemed Cdc14 an attractive potential target for several reasons. First, when Cdc14 orthologs were inhibited in other plant pathogenic fungal species, inactivation eliminated pathogenicity. Second, the catalytic domains of Cdc14 orthologs are highly conserved, so it can be hypothesized that we can use the known structure of the Cdc14 ortholog in, say, budding yeast or S. cerevisiae as a model to design a competitive inhibitor in F. oxysporum. Additionally, because the inhibition of CDC14 hinders multiple central processes, increasing the likelihood of cell death, this theoretically decreases the chance that the pathogen develops antifungal resistance to the inhibitor. And finally, while conserved in fungi and animals, the CDC14 phosphatase genes do not exist in higher plants. This ensures that my novel inhibitor will not have adverse effects on the agricultural products it was designed to protect. Ultimately, the purpose of my project was twofold. First, to determine if it is appropriate to use the known structure of SC-CDC14, which is the ortholog in Saccharomyces cerevisiae, as a template for modeling FO-CDC14, which is the ortholog in F. oxysporum. And second, to develop a novel fungicide inhibitor of FO-CDC14 and eliminate the pathogenic properties of F. oxysporum, with the overall goal of increasing global food security and avoiding antifungal resistance. So to begin this project, only the raw genome sequence of an uncharacterized pathogenic protein from F. oxysporum was known. This was converted to an amino acid sequence using GenScan. To hypothesize the function of this protein, an NCBI protein blast search was conducted. A domain hit output by searching against S. cerevisiae suggested that the protein was likely part of the CDC14 family, as demonstrated here in figure 1a. To further this hypothesis, a multiple sequence alignment between several eukaryotic species whose orthologs were more extensively characterized was carried out using the Clustal Omega program. And the red box in figure 1b represents the catalytic domain of the protein, of which the sequence is conserved across all species. Following this hypothesis of the F. oxysporum pathogenic protein as a CDC14 phosphatase, I used the SnapGene Viewer program to construct a plasmid for use in the recombinant cloning of the gene. The introns from the original blast sequence were removed, and the DNA coding sequence for FOCDC14 was ligated into the plasmid in a double digest reaction. Here's a map of the fusion plasmid, though in the interest of time, I'll refrain from going into further depth into plasmid design. Next, E. coli transformation, followed by affinity chromatography and SDS page, were conducted to express and purify the CDC14 protein. Figure 2b displays UV absorbance of the flow-through solution as the proteins pass through the affinity column. This data, coupled with the SDS page data in figure 2c, allowed me to conclude that the purification of the FOCDC14 protein in the lab was successful. So after expression, purification, and concentration determination of the target FOCDC14 enzyme, steady-state assays were used to identify the function, efficiency, and specificity of FOCDC14. The activity of FOCDC14 as a phosphatase was confirmed by a phosphatase assay, defining the class of its likely substrates as phosphopeptides. The concentration of PNP produced from dephosphorylated PNPP was significantly lower with the addition here of sodium tungstate, which is a known inhibitor of phosphatases, as seen here in figures 3A and 3B. The catalytic efficiency of FOCDC14 for PNPP was determined in a steady state kinetic assay for later comparison to inhibitor kinetic assays. FOCDC14 Kcat and Km values were calculated and recorded in figure 4B. The hyperbolic shape and high R-squared value validated the data as it indicates that the mechanism conforms to Michaelis-Menten kinetics. Additionally, selectivity of FOCDC14 for phosphoserine peptides with adjacent proline and basic residues was identified in a substrate specificity assay with 24 different small phosphopeptides. These findings would help to design an inhibitor with similar structure to biological FOCDC14 substrates. For 3 of the 5 most readily catalyzed peptides, the addition of a basic residue in the P+4 to P+6 region was the defining characteristic. Altogether, these steady state assays and kinematics confirm the function, efficiency, and specificity of my FOCDC14 phosphatase, allowing me to next select and optimize a lead inhibitor using in vitro and in silico methods. So here we go. From the amino acid sequence of FOCDC14, a homology model of the enzyme was developed in the Mo software for use in inhibitor optimization experiments. The model was developed using the known structure of S. cerevisiae. RMSD coloration in figure 6A demonstrates strong structural similarity between SC-CDC14 and the homology model, particularly at the active site, revealing that the model is plausible. A Ramachandran or phi-psi plot revealed a relatively low number of residues containing undesirable torsion angles, further affirming the validity of the homology model was executed in Mo for comparison to later inhibitor docking results. And the 2D interaction map confirmed several non-covalent contacts between the substrate and homology model's catalytic active site. After generating the homology model, ligand model docking and inhibitor screening data were used for the selection of a small molecule as a competitive inhibitor for FOCDC14. 16 lead inhibitors were docked in the binding site of the homology model in Mo, and the same 16 were used in an in vitro inhibitor screen assay. Of the 16 molecules, inhibitor G6, shown in figure 7E, was chosen as the lead inhibitor because it demonstrated a low S value when docked into the homology model, formed the same interaction seen in phosphopeptide substrates of the FOCDC14 ortholog, and demonstrated high percent inhibition of FOCDC14 in the inhibitor screening assay. So finally, to increase the binding affinity of this G6 inhibitor to FOCDC14, modifications were made to optimize the inhibitor using the builder in Mo. Multiple factors were considered during the modification process, including H-bond donor and acceptor regions, torsion angles, molecular weight, access to various active site binding pockets, and more. Following the strategic addition of multiple functional groups, the binding score of the optimized competitive inhibitor improved from negative 5.83 to negative 9.40 kilocalories per mole. Pictured here is the final optimized competitive inhibitor and its many contacts with the active site of the enzyme. Now, before discussing the larger implications of this project as it relates to One Health topics, let's revisit the hypotheses I put forth. First, after obtaining results from the protein blast and multiple sequence alignment, I hypothesized that the pathogenic F-oxysporum protein was affirmed by the phosphatase assay. Then, I hypothesized that a novel inhibitor based upon the G6 molecule can competitively inhibit FOCDC14. This hypothesis remains inconclusive until my optimized G6 inhibitor is synthesized and tested in vitro and in vivo. Nonetheless, the project made notable progress in characterizing FOCDC14 structure and ligand interaction such that future studies may draw from my work in an effort to fully develop an FOCDC14 inhibitor. Molecular modeling and assay results were especially promising, setting the stage for future studies on that topic. The results of this project have broad implications, touching on a variety of pertinent One Health topics, including food safety and security, antifungal/antimicrobial resistance, and emerging infectious diseases, as well as the general overlap of health threats shared by people, animals, and the environment. As a plant pathogenic fungi, Fusarium oxysporum has jeopardized food security and safety in developing nations and devastated agricultural economies the world over. Yet I'm optimistic that the biochemical and computational approaches explored in this project present major progress in combating this global health issue. So to conclude, I would like to thank everyone, especially the panel of judges, for their time and consideration. And thank you to everyone who made this symposium possible during these uncertain times. I really appreciate it. Thank you, everyone.